Embryology of the female external and internal genitalia. The development of the female reproductive system involves complex processes during embryonic and fetal life. The differentiation of external and internal genitalia is influenced by genetic factors, particularly the presence of certain sex chromosomes and hormones. First, general overview of sexual differentiation. Chromosomal sex determination. At fertilization, the chromosomal sex of the embryo is determined. Females have two X chromosomes, double X, and males have one X and one Y chromosome, XY. The presence of a Y chromosome leads to the development of male characteristics, whereas the absence of the Y chromosome, double X, leads to female development. Gonadal development. The gonads, ovaries in females, begin developing around the 5th week of gestation. In females, double X, the gonads develop into ovaries. In the absence of the SRY gene, found on the Y chromosome, which stimulates the development of testes in males, the default pathway leads to ovarian development. Second, development of the female internal genitalia. The female internal genitalia develop from the mesonephric ducts, also called Wolffian ducts, and the paramesonephric ducts, also called Mullerian ducts. These ducts are initially present in both male and female embryos. Mullerian ducts, paramesonephric ducts. In the female embryo, the Mullerian ducts develop into the internal genitalia, uterus. The two Mullerian ducts fuse to form the uterus, which consists of the fundus, body, and cervix. Fallopian tubes. The upper part of each Mullerian duct forms one of the fallopian tubes. Upper 2/3 of the vagina. The lower part of the Mullerian ducts forms the upper part of the vagina. These structures develop under the influence of estrogen produced by the developing ovaries. Wolffian ducts. In females, the Wolffian ducts regress due to the absence of the male hormones, androgens. In males, the Wolffian ducts develop into the epididymis, vas deferens, and seminal vesicles, but in females, they do not contribute to internal genitalia. Third, development of the female external genitalia. The development of the external genitalia, vulva, in females also involves the differentiation of structures from the genital tubercle and genital folds. Genital tubercle. The genital tubercle forms the clitoris in females. It develops around the 6th week and becomes prominent as the clitoris, a structure that is homologous to the male penis. Labioscrotal swellings. The labioscrotal swellings, a pair of bulging tissues on either side of the genital tubercle, differentiate into the labia majora in females. In males, these swellings form the scrotum. Urethral folds. The urethral folds, located beneath the genital tubercle, fuse in females to form the labia minora, the inner folds of the vulva. In males, these folds fuse to form the shaft of the penis. Vaginal opening. The vaginal opening develops from the urogenital sinus, an area that forms the lower part of the vagina. This area also contributes to the formation of the urethra. Key hormonal influences. Absence of testosterone and anti-Mullerian hormone, AMH. The default female pathway is initiated in the absence of testosterone and AMH. The absence of these hormones allows the Mullerian ducts to develop into the female reproductive structures, uterus, fallopian tubes, and vagina. Estrogen, produced by the developing ovaries, plays a key role in the differentiation of the internal genitalia, particularly the uterus and upper vagina. Summary of embryological development. Internal genitalia. The Mullerian ducts develop into the uterus, fallopian tubes, and upper vagina, while the Wolffian ducts regress. External genitalia. The genital tubercle becomes the clitoris, the labioscrotal swellings become the labia majora, and the urethral folds form the labia minora. Timeline of development. Week 5 to 6. Development of gonads, ovaries or testes. Week 6 to 7, formation of the external genitalia. Week 8 to 10, differentiation of the internal genitalia. Mullerian ducts form uterus, fallopian tubes, and vagina. Week 12, external genitalia fully differentiated. Week 16, development of the ovaries and the uterus is recognizable.